You know you're going to see exam questions about the classic genetics experiments. And if you want to understand Griffith's transformation experiment and get those points, you need to understand the one result that makes zero sense. So let's take a look at that. Frederick Griffith is working on developing a vaccine for pneumonia. Pneumonia is caused by a bacteria. We now know it as Streptococcus pneumoniae. And Griffith found that many infected patients had multiple different types of this bacteria in their systems. Type 1, type 2, type 3. The different types have different cell surfaces, so the infected person's immune system generates different antibodies against them. Think of it like the same bacteria, but wearing different outfits. Scientists at the time thought that these types were unchangeable, so a type 1 cell would always be a type 1 cell and produce more type 1 cells. But Griffith wondered whether one type could convert into another, something useful to know for his vaccine research. You see, bacteria like these, capable of aggressively infecting and causing serious illness, we can describe as virulent. Something about their biology allows them to successfully invade and multiply. And for these pneumonia-causing bacteria, that's a polysaccharide capsule. Polysaccharides are sugars. This creates a sort of jelly-like protective surrounding that helps the bacteria stick to surfaces and makes it harder for immune cells to engulf the bacteria. Bacteria with a capsule form large, smooth colonies in a petri dish. So we call them smooth or S-type. Remember, type S makes you sick. But sometimes mutants would appear in those petri dishes that lacked a capsule. Their colonies look smaller and rough. These rough or type R bacteria are nonvirulent or avirulent. They don't cause pneumonia. A mutation breaks a gene needed for building that capsule, and without a capsule, they're more easily dealt with by defensive cells. So can one bacteria type convert to another? Well, Griffith used two types, virulent 3S and non-virulent 2R, to find out. He injected mice with type 3S bacteria. Type 3S, S, smooth colonies, sick. This is a pneumonia-causing type. So what happened to the mice? Unsurprisingly, they died of pneumonia, and autopsies showed they were full of type 3S bacteria. The second group of mice were injected with type 2R bacteria. Did those mice develop pneumonia? Not from a rough non-virulent type. Their immune systems were able to fight those bacteria off. A third group of mice were injected with heat-killed type 3S. So the same bacteria from the first group, but dead. Do dead bacteria cause pneumonia? Apparently not. The mice survived and no living type 3S bacteria were found in their systems. And the fourth group of mice were injected with heat-killed type 3S and living type 2R bacteria. We saw those separately, and the mice were fine in both of those scenarios. So we expect, no, they're all dead from pneumonia. Autopsies found living type 3S cells, just like the dead ones that were injected, but they're alive now? Okay, but how? Well, maybe the smooth bacteria weren't all dead, and some live ones slipped into the procedure. But that third group with just heat-killed 3S was a control for the sterilization procedure. No living 3S ever showed up in those mice. This bacteria is heat sensitive, and heating to 60 degrees Celsius for a few hours ensured no living bacteria remained. Well, maybe the living 2R bacteria just mutated into 3S bacteria. After all, they already knew spontaneous mutations could change smooth bacteria to rough, 2S to 2R, or back, but Griffith controlled for that possibility too. In the second group of mice injected with just 2R bacteria, there was no conversion to 3S. None of the mice got sick. So this wasn't random. 2R bacteria didn't spontaneously become 3S all on their own, only when they interacted with 3S material. All right, well, that means that some material in the heat-killed 3S cells transforms non-virulent type R into virulent type S. Griffith called this material the transforming principle. But what was it? Well, he knew that it was something that could survive the heat treatment. He thought it might be part of the capsule. Whatever it was, the transforming principle was heritable. The transformed 3S bacteria reproduced to make more 3S bacteria. The transforming principle was passed from one generation to the next. Cool, but what is the transforming principle? Well, Griffith didn't solve that mystery. He was killed during the London bombings of World War II. But just a few years later, other research teams would build off Griffith's work with more classic genetics experiments to show that the transforming principle, the molecule of inheritance, is DNA. Well, we know now that through a process we call bacterial transformation, the living 2R bacteria took up fragments of 3S DNA from the dead cells they were mixed with and incorporated those 3S genes into their own chromosome, allowing them to produce polysaccharide capsule, transforming harmless 2R bacteria into virulent 3S bacteria. Drop me a thumbs up if we've cleared up the Griffith experiment. Subscribe for more biology content from me. Check out my website for tutoring resources, and I'll see you next time. Bye.